Hello, my name is Marina DiStefano, and I'm a clinical molecular genetics fellow at Harvard and a postdoc in the ClinGen resource. This is part 1 of a 3-part training series on transcript annotation and curation. Part 1 will discuss transcript annotation. This is an outline of the entire series. As I said, in this first part, I will cover why transcript curation matters, citing examples from the hearing loss clinical domain. I will also provide an overview of transcript annotation bodies and their differences. Please continue on to Part 2 for more discussion on transcript curation resources, and Part 3 to hear about a specific transcript curation strategy that we deployed in the hearing loss domain. As many biocurators already know, gene curation is required for variant curation. If a gene is not associated with disease, the effect of a variant on that gene cannot be determined. This is best represented by the graph on the left here, modified from Daniel MacArthur, with variant-level evidence plotted on the y-axis and gene-level evidence plotted on the x-axis. You can have a very well-characterized variant and a well-characterized gene on the top right, such as delta F508 in CFTR, which is definitively associated with cystic fibrosis. You can also have a less characterized variant of uncertain significance, or VUS, in a well-characterized gene on the bottom right. You can even have a VUS in a GUS, a variant of uncertain significance in a gene of uncertain significance on the bottom left. But you can never have a variant with significant evidence in a gene with little to no evidence. I am hoping in this talk to convince you that transcript curation represents an additional level of curation required for variant curation. Transcript curation is critical for variant interpretation because transcript choice could alter the molecular consequence of the variant, and I will provide examples of this. Clinical labs often choose the most severe molecular consequence for interpretation, which may not be the most biologically relevant. Transcript choice can vary depending on the disease being evaluated and the tissue expression profile of the transcripts. Clinical labs also often choose the longest transcript for variant annotation, which may not be the most biologically relevant choice, depending on tissue distribution of the transcripts. This is an example from the hearing loss domain that supports the idea of the need for transcript curation. TBC1D24 is a gene that is associated with non-syndromic hearing loss, Doer's syndrome, which is a syndrome characterized by deafness, nail abnormalities, bone abnormalities, and seizures, and a spectrum of epilepsy conditions. As you can see, RefSeq, a transcript annotation body which we will talk about shortly, lists 2 curated transcripts for this gene. The top one is expressed in mouse neurons and contains an extra exon 3. The bottom one is expressed in mouse cochlea and non-neuronal tissues. In the literature, a frameshift variant was identified in this unique exon of the transcript that is solely expressed in neurons, leading to a loss of function of this transcript. The individuals that harbored this variant had severe lethal epileptic encephalopathy without hearing loss, which is supported by the tissue-specific expression in mouse. I'll now take some time to discuss the most commonly used transcript annotations. The first set is the Ensembl and GenCode set, which is annotated by NCBI. This was previously largely done in an automatic fashion, but Ensembl curators have informed me that more and more manual curation is being used for these transcripts. They are pulled and aligned directly to the genome build. As I'm sure you've seen before, they are labeled with the ENST label. ENSP is for the translated protein product. It's important to know which curation references use these transcripts. The most critical ones are the reference population databases, GNOMAD, EXAC, and the 100,000 Genomes Project. GTEx, which I'll discuss more in video 2, is a very useful transcript curation resource and also uses Ensembl transcripts. Decipher and Cosmic use them too. This is a screenshot of the Ensembl website. You can see each transcript listed along with the corresponding RefSeq transcript, which is a helpful feature for curation. The flags listed at the end are evidence flags for each transcript. If you mouse over each tag, it will specify what each one means. For example, TSL1 is Transcript Support Level 1, which means it is supported by at least one non-suspect mRNA. RefSeq is the transcript annotation effort from NCBI. These transcripts are designated by curation and are independent of the genome build. This can mean that the genome build and RefSeq can differ. This can pose a problem because clinical pipelines often call variants off of the genome build and then annotate them with RefSeq transcripts. RefSeq gene is the most thoroughly curated set of RefSeq transcripts. The two prefixes for these transcripts are N and X. N transcripts are supported by some evidence, either literature or GenBank cDNA data. GenBank is a database hosted by NIH that contains sequences from 260,000 species collected from individual labs and batch submissions from large-scale sequencing projects. The second letter in the prefix denotes the type of transcript. M is for mRNA, R is for non-coding RNA, and P is for protein. So, for example, the most common prefix you see is NM, which means it is a curated mRNA transcript. RefSeq is most commonly used by clinical labs and research publications, which is important to note for biocurators. This is a screenshot of different parts of the RefSeq gene website. It was too large to capture it all, but I've included useful parts. The top is the gene level summary. Below is their genome browser that lists all of the RefSeq transcripts. Further down on the website, each transcript is listed with a short explanatory sentence. These annotations are pulled into Element if you use that genome browser. The top right is a picture of the bibliography for the gene. I've been informed by RefSeq curators that these references are a random listing of all of the references, so it is important to click and evaluate all of the references. There is also a graph of RNA-seq data. I personally prefer the exon level expression of GTEx, but this set is also useful. I'm now going to talk about the Ensembl EBI and NCBI collaboration efforts. The first effort that was developed was the Locus Reference Genomic, or LRG effort. These transcripts are highly curated, and their purpose is to ensure efficient and consistent variant reporting by choosing a clinically relevant transcript. This usually corresponds to one transcript per gene and is relatively stable. As of a few weeks ago, there were only 1,200 or so LRGs, indicating that it is a time-consuming effort. LRG has curators, but it also solicits transcript curation from content experts. For example, we have curated 100 or so hearing loss genes, and we worked with LRG to submit those curations and generate LRG transcripts for all of those genes. This is the workflow that the LRG effort used for incorporating our hearing loss transcript curation. As a part of this effort, NMs and ENSTs are 100% harmonized, and an LRG is created using these harmonized transcripts. This effort will feed into the main effort, which I'll discuss shortly. This is the display of one of the hearing loss transcripts we curated. You can see the harmonized and mapped RefSeq and Ensembl transcripts. The newest collaboration from Ensembl EBI and RefSeq is the matched annotation from NCBI and Ensembl EBI project, or the main project. This is a collaborative transcript set, including the best that RefSeq and Ensembl offer. The transcripts are aligned to the newer genome build GRCH38, anticipating that clinical labs can switch to this set when the population reference databases switch to this build. Transcripts are fairly stable, highly curated, and have 100% identity between annotation efforts. The main select set is the first set to be released, which includes one transcript per locus, and 50% of these transcripts were released in December last year. The remainder will hopefully be released later this year. The second set is the main plus set, which continues on with the LRG project, providing highly curated transcripts for particular user groups like clinical labs or researchers. These will be completed in the future. Ensembl EBI and NCBI recently had a very nice explanatory webinar on this effort, and I highly recommend reviewing the slides from that webinar or the recording for more information. The 50% of the main select transcripts are available as a download on NCBI's FTP site. In conclusion, I hope that I've convinced you that transcript curation is critical for variant interpretation. I've explained the existing annotation efforts and how they differ, but they are in the process of harmonization. Please check out my other two recordings to find more information about curating transcripts. Thank you.